all the panelists. And I think we have one more person, Kate, to bring up, or at least she's not on my screen, but if we could include her on the screen. But one of the, is Kate, I see Kate as a little, maybe it's just the way my screen is oriented. But, there, now I see Kate as well on the group of five. So one of the points, that the major goals of this symposium is to really help people get guidance on what they should do when they are trying to map M6A and study M6A dynamics, because I think that's probably the thing that people really are trying to understand. And, I want to think a little bit about how Schraga's method, theories impact our understanding of dynamics. But, right now we're really talking about three methods, which are probably in my opinion, the most innovative methods right now. But but the advantage of ETAM-seq and Glory is that you can get really good quantitative information. So my question for the panel and include, is which one should they use? Should you use ETAM-seq or should you use Glory? What's the best one? If some person comes to you, and let's say you didn't invent ETAM-seq or you didn't invent Glory. If some person comes to you and says, hey, look, I want to understand how M6A changes in muscle development, what method should they use? And, why don't we start with Weixin? What do you think? What would you tell them? And why that method? And, we're all friends here, but why not Glory, or why not Dart-seq, or why not, other methods?
If the target system is compatible with genomic manipulation, I think Dart-seq would be a very good method. You get super high sensitivity and it's compatible with canonical RNA-seq workflow. I have to admit that with an actual deamination step, your RNA-seq workflow is not as easy. In terms of the two deamination methods, I do think that right now with TAT 8.20, our deamination rate is not as high as Glory. So in terms of control-free detection, I would perhaps recommend the chemical method. I think our method is particularly good with low input materials since we don't see much degradation. We have collaborators who approached us say that we only have 3,000 samples, 3,000 cells or 5,000 cells. Do you think we can use your method? I think that would be a scenario that ETAM-seq would be particularly useful. Also, site-specific detection. I think that would be useful.
What is the major reason? What is the major problem with ETAM-seq that you would say that you should keep in mind, and that would influence your choice of whether to use ETAM-seq? And I would love to hear what Chengqi thinks about the major problem with Glory, Kate, what she thinks about the major problem with Dart-seq, and Schraga, I would like to hear his opinion. He invented Master-seq. I'd like to know if there's a role for Master-seq and even any of our old methods, the MIRIP-seq2, our M6A-seq. But what's the biggest drawback of ETAM-seq, or is it the best thing since sliced bread?
I think we have no intention to replace our other methods. One obvious limitation here is that we don't get enrichment. So prepare to pay more.
So you because you mean that you do a lot, there's a lot of sequencing depth required to get the data you need.
You can't really do M6A enrichment. You're not sequencing a subpopulation of the transcriptome. You're sequencing the entire transcriptome to extract that 0.2 to 0.6% information. So their cost would be a concern. Of course, you can always do immunoprecipitation, then apply the method, but you lose the benefit of quantitative detection. So that would be a huge drawback as well.
I think it's more important to get quantitative detection than it is to just know if M6A is there, because I think there's a big difference between an M6A that's 10% methylated and 90% methylated. And so when you use the enrichment methods that Schraga and I and Kate developed many years ago, that immunoprecipitation can immunoprecipitate a very low abundance transcript or a very low stoichiometry M6A, which is not very meaningful. What about you, Chengqi? What do you think are the major limitations of glory? When should you not use glory, and when should you use glory?
Right. I think I will start with current drawbacks we already see with glory. Glory fragments RNA, it degrades RNA. The concept, I think, similar to bisulfite to DNA, but although it doesn't, degrade DNA that severely compared to the original condition of bisulfite to DNA. So to extend this statement, I think, let's say, I think Weixin certainly has a great example of applying ETAM-seq to the deep sequencing free methodology, right? The enzyme is mild, so it does not degrade RNA. Currently, if we use the current condition of glory, it fragments RNA into shorter fragments. So if we're one is interested in identifying the methylation for a particular locus, particular site on mRNA transcript, first of all, we're looking at ternary base composition, right? Since glory, turns majority of A into G, so there's only M6A left. So it's not easy to design primers. That's first thing. Secondly, there's degradation, so the efficiency can be low. So that I certainly want to point out, and I want to make it very transparent to the scientists, to the entire field, that I think currently it degrades RNA and it's not good for let's say Sanger sequencing to integrate the potential locus for M6A. We're working on that. We try to improve the methodology so there's less degradation, and we can work with smaller amount of samples, let's say 1,000 cells or even lower. So that's ongoing. We have a different condition we have to improve. So that's, I think, the current major drawback of glory. And I think for the potential advantages of glory, since the small molecule it really doesn't have any potential sequence bias. So potentially, I'm certainly very looking forward to Weixin's newer version of the TEDR. But for small molecules, in principle, it doesn't have any sequence preference. So I think for this property, the quantification power could be more uniform across different sequence contexts. Although there is a strategic motif for M6A, but I think if we further extend M6A, let's say into metal 16 and even additional sites, I think a more uniform quantification probably is also very important. And this is, I think, what we already see for glory. So that's
So
yeah,
we've
go ahead,
tried
Samie.
we've tried both ETAM-seq and glory in our lab. Glory is a lot easier to do because we can just buy sodium nitrite and start doing the experiment. It works great in my opinion, but there's a lot of fragmentation. And so we definitely do size selections of RNA before we even sequence it. And it's kind of better in our view to have lots of input RNA because a lot of it's going to get degraded. But it does work nicely in terms of converting the A's to G's in the sequencing, except at the M6A level. For us, I think the challenge is the amount of degradation and the amount of input material that you have to have. But the other thing is, I think the every fragment that you get from glory is a short fragment. We're lucky if we get a 40 nucleotide long read out of glory, and that necessitates really high sequencing depth to cover the transcriptome, because we normally do 150 nucleotide long reads, but we're only giving it a 40 nucleotide long read, so we're missing out on extra reads. I think you had 100 million sequencing read depth in your experiments. Is that right, Chengqi? Is that accurate?
So first of all, of course, you get some more M6 sites and more accurate quantification with higher sequencing depth. That's true. First of all. Secondly, there's degradation problem we already mentioned. Thirdly, we're improving on that, and we have progress. So we look forward to certainly have higher. Longer sequencing inserts as well as lower starting input. So that's ongoing. But to answer your question, currently why we normally there's also a short insert, something like 40 to 60. It also it is also affected by the current library preparation protocol. So let's say eClip, basically, or even we tried. I think we tried different library preparation methods. eClip works. Small RNA library preparation methodology works. But let's say for these two, the average length of the insert, that changes as well. So there is fragmentation, but the shorter reads is also partially because of the library preparation procedure. We have a Nature protocol paper under revision. So at least I think we try to be transparent. We try to make sure these situations. But on the other side, I think for future, yes, improved condition to have lower degradation, to have smaller degradation and smaller input sample would be good.
Yeah. So the enzyme, our experience with eTAM-seq, it really depends on when we can make the enzyme, and making the enzyme and keeping it stable, that's a little bit tricky. So it's been hard for us to do global transcriptome wide. We may ask you for some of the enzyme just to compare it with the enzyme that we make. But if the enzyme is super stable and abundant, it seems like it's very advantageous to use eTAM-seq, because you're going to have to pay for getting quantitative information, because you're going to have to pay for it either way. You're saying, Weixin, that the problem is you need a lot of sequencing depth. You need a lot of sequencing depth with Weixin's method, with eTAM-seq, or with Glory, and with Kate's method, a lot of it is single cell now. That's extremely expensive. So, I think that it's tough to choose these methods, but I definitely like getting longer fragment reads with Weixin's method, but the barrier is you have to write an email to Weixin to get the enzyme. Kate, what do you think
or
about?
Weixin can find a vendor from the audience.
Yeah, I hope someone from NEB or some other companies listening. Kate, what about DartSeq? And one of the things that was really, and I should mention, people feel free to ask questions in the Q&A box, and we'll try to get to them as well. But, Kate, I'm wondering, DartSeq, obviously one of the powers of DartSeq is especially with the long read data, is you might be uncovering completely new modes of methylation regulation, maybe bursts, like some transcripts, there's no methylation at all, regardless of the EJC. And then the methylation complex happens, and then all the transcripts are methylated, and that might explain individual. But what do you think is the right use for DartSeq? What's the right use for in-cell DartSeq, in animal DartSeq, and in vitro DartSeq?
Yeah, so good question. I think some of the main limitations of DartSeq are also its strengths, right? So it uses expression of this ApoBec-YTH protein, which is very convenient because you can just use any RNA-seq library method prep of interest, and you don't have to worry about, huge, enormous coverage if you're just doing a bulk RNA-seq experiment. Single cell is a different story. That's just inherently more expensive. But it makes it very easy. And as you said, Samie, it's compatible with long read sequencing. Very early days, we published it with PacBio. I think coupling modification knowledge with single molecule sequencing is going to be really powerful going forward. So those are some of the advantages that make DartSeq great. At the same time, it involves expression of this ApoBec-YTH enzyme, which binding to all these thousands of M6A sites in cells and creating these CDE mutations, which, what's that doing to the biology of the cell? And ultimately we do this because we care about the biology. So that's an admitted major limitation, right? So in our lab, we frequently, if we're using cultured cells, we'll express or induce the enzyme for the course of maybe 24 hours. The mice, they're designed to be an inducible system for that reason. We were worried that they wouldn't even survive. I mean, the tamoxifen injection is done over the course of like a week, and then we wait another week. It's like a 5-day injection, and then we wait a week to sack them. I, for one, was honestly surprised. They have no notable, physical, abnormalities. We've done, compared RNA-seq profiles to them and wild-type mice, and we don't see any huge changes. So the mice seem to do okay. But all that said, again, you're expressing this enzyme that's making these edits, so you have to be cognizant of what that's doing. So to answer your question, Samie, I think if you're worried about that, that's where in vitro DART-seq would be advantageous, right? Because there you have an RNA sample, and that could be something like a human sample where you're not going to be expressing an enzyme in a human sample, or a patient sample or something like that, and you can just mix it with the enzyme in vitro. So there it becomes very powerful. That said, I think the mice can be great because, obviously we can study these effects of, a stress or a, learning and memory, or like any, you name it, right? And you can study it in different tissues. So that I think is a very powerful tool now that we have as well. So I guess the short answer is it kind of depends on your application, but the main limitation, I think, for DART-seq is expression of this protein that's making these edits. So you always have to be cognizant of that. And admittedly, it doesn't do as precise of a job as, GLORY and ETAM of quantifying M6A. That's still something we're trying to work towards. So I think those are the main limitations, but it certainly has its advantages as well.
But one of the things that I worry about with DART-seq is, to what degree is editing a reflection of M6A stoichiometry versus to what degree is it a reflection of other things that are bound to that same M6A site, or let's say the ribosome knocking off the enzyme, because this, the in vivo stuff, the cell-based and the in vivo is in living animals. And, I wonder if some of the stoichiometry or some of the changes in reactivity is because, these mRNAs are now in granules and they're not accessible, or these mRNAs are now being translated more efficiently. And so what do you think about that in terms of, you're definitely seeing lots of variability in the editing, and it's very fascinating, but there is correlations with stoichiometry, but how much of it could be due to other things? Because the in vitro methods of Weixin and of ETAM-seq and GLORY, there you don't have to worry about competing proteins.
Right. Yeah. And that kind of speaks to, this inherent limitation of expressing this enzyme, right? So there are some things that we can't know, right? We don't know the precise things that might influence accessibility of an M6A site, because like you mentioned, there's the ribosome, are there RNA binding proteins, or the activity of ApoBec, right? That could be influenced. There might be structural elements that maybe the M6A is accessible, but maybe somehow the cytidine isn't easily accessible to the ApoBec part of it, right? One thing we have done is we've compared cellular DART-seq to in vitro DART-seq. So with in vitro, right, we're just using trizol to extract the RNA from the same cells. So that removes other RNA binding proteins, that removes, the ribosome, et cetera, and we see pretty good overlap. So that suggests that, in cells, the enzyme or the M6A sites are pretty accessible to this enzyme, the same that they are in vitro. So that gives us, that was very promising for us to see. So yeah, but like you said, some of these things are unknowns, and we don't exactly know how different cellular contexts or locations of the RNA in the cell might influence the accessibility of those M6A sites.
So that's great. Veronica de Jesus wants to know how these methods compare to nanopore, and should she use these methods or nanopore? But I think, Kate, in the panel discussion tonight, maybe you can address that when we go through all the nanopore, because I think there's that question.
We'll definitely be talking about that question, Veronica.
Yeah. But Schraga, you are from the very beginning in M6A, you made the M6A-seq method, which is basically the same as our MIRIP-seq method, but then you improved it in so many interesting ways. You made MIRIP-seq2. Is there any value to these older methods, which are still the methods? Every time I get a paper alert on Google, I still see people using these older methods. Are there any advantages to your old methods and our methods? Or should everybody be switching to these new methods? What are the pros and the cons from your perspective?
So first of all, I think ETAM-seq and Glory, they're really, really game changers. They allow the kind of resolution we've all been dreaming about for years, the quantification we've been dreaming about for years, something we could kind of sense but never really see. And for the first time, they're allowing us to measure. So they're absolutely great. And I think especially if you compare these two methods, like we developed in the past for quantifying M6A with MasterSeq, I think basically I don't see a place for MasterSeq anymore these days with the availability of these methods. MasterSeq has its biases and its limitations to certain consensus motifs, and I just think Glory, which we also tried out in our lab, I think it overcomes these methods. And Weixin, I will definitely take you up on your offer of also trying out the EDZEB. We didn't try the method because of that limitation, but we would be delighted to try it out as well. What I think is important, though, to consider, and it kind of goes back to the point I was making, is as a field, I think we need to consider what resolutions of M6A are actually interesting or the most relevant one from a biological perspective to quantify. To what extent do we need to care about yet another one of 12 M6A sites per genes and about getting its quantification precisely right? Or is what we care about much more is the overall load of M6A per gene, so some index that would integrate the overall M6A level in a gene? And I would argue that if we think that the biological consequences of M6A do not reflect any individual site here or there, but instead reflect the cumulative weight of M6A load over the entire gene, that then maybe what we want is a method that can capture that. And I think when it comes to that, M6A-seq2, which we published at the time, that's actually one thing it offers, and which at a relatively limited read depth, because you do the enrichment, you can get these kind of profiles, and which will, for example, beautifully correlate with with RNA stability to an extent that if you were to take just individual sites here or there, it's simply something hard to recapitulate. So we've actually we've analyzed some of the Glory data, and we've tried to see to what extent can we reach the same if we just kind of try to develop a similar kind of index on the basis of this data. And what we can see is that as you have more and more read depth, you can get closer and closer to these profiles that you can readily get with the simple M6A-seq2 experiment. But the kind of depth that you need there, as we're all talking, you need to get to depths of about 200 million reads, and even then you can get this kind of depth and indexes for highly expressed genes. Whereas if you go for more lowly expressed ones, you're still going to be more limited. So I think these inherent limitations, the kind of depth that you require to actually accurately estimate individual M6A sites, are going to be a limitation for these kind of utilities. And depending on what you want to know, so what's important, do you care about tying M6A to downstream processes, or do you care about understanding what gives rise to different levels of M6A at different sites? These are very different questions, and depending on that, I think there is utility for the one or utility for the other. So I still think M6A-seq in its classical incarnation definitely has its utilities. The fact that you can multiplex it readily, the fact that with low coverage you can get these rich landscapes, which ultimately converge also with the ones that we can get with Glory. All of that, I think, still leaves it as a relevant method, but again, of course, depending on the application.
So let me say, I agree with you 100%. I think that MasterSeq, which is a method that uses the MASF ribonuclease, and that ribonuclease is sensitive to M6A in very specific sequence contexts, was very great at the time, but now we don't necessarily need it. But I also agree with you that M6A load is the concept that people should use. I know there are papers that claim that this M6A site does this and this M6A site does that. And there may be some individual mRNAs where a M6A was evolved to have some very unique function, but it's the overall weight of M6A that's going to determine the degradation. So we've taken a lot of Chengqi's data and Weixin's data to calculate M6A loads, just like you have. And it does make an even stronger correlation with degradation like you're finding. But I agree with you. I think this is the issue that that we're sort of encountering a little bit, which is that a lot of sites are based on a small number of reads. And let's say it's predicted to be 50% stoichiometry. Well, maybe a few more reads, it could go down to 20% when you get enough read information. And definitely the MIRIP-seq2 the M6A-seq2 method, where you can get a lot of reads and get a very good measurement of the load, might be a good way to assign it. But I think part of the solution is to get deeper read coverage on your GLORY and ETAM-seq, and then you might get a very a good M6A load.
I'll just say the solution is not as trivial as it sounds, because I think you need to balance it also with complexity. And I'd love to hear kind of Chengqi's equations, but at least for GLORY, the complexity that we've been able to get has not been infinite. And that just means that even if you were to sequence deeper and deeper, the return in that would rapidly start decreasing. So you wouldn't really, it's not just taking a sample and just, sequencing a full flow cell out of it, and you just get more and more sites. I think even with the published data, I think the complexity, in terms of the duplication or the actual complexity, I think there's about, if I remember correctly, about a 70% duplication rate. And that just means, you cannot at some point sequence it deeper and still get more information. I'd love to hear kind of Chengqi's thoughts on this as well, and in terms of what kind of complexities they're able to reach. Again, it's a beautiful method. I just kind of, in terms of thinking of its limitations, I think that's one limitation.
What do you guys think, Chengqi and Weixin?
So maybe I have some information from my side to the question. So. Thank you, Schraga, for mentioning this. And I think Samie also mentioned earlier that we indeed sequenced very deep for one particular data set we have in our GLORY paper. But I think the purpose there is not to try to get as many M6A sites we can get. I think the concept there is exactly as what Schraga has just mentioned. We want to see when it gets, it reaches this plateau. And we also, from a very deep sequenced data, we also do downsampling and to see whether, how many genuine MC sites we can recover. So currently, I have to admit that the bioinformatics algorithm we use currently is certainly not optimal. So basically, the threshold is, let's say artificial. I don't remember the exact number, but let's say we require every single A site to be covered by let's say 50 times, and we also disregard, let's say, those A sites which has only, let's say, two remaining A's. That means, first of all, GLORY currently at this moment, to be more accurate, we disregard those modification sites. Those are very low, let's say below 5%, but one can also argue that those low modification sites could have less function, but one could argue this, all right? So I think currently there's certainly room to further improve the bioinformatics algorithm with limited sequencing depth. We do not, we don't encourage to sequence as deep as possible. But, what is the best sequencing depth? I think, and what is let's say, the redundancy, what is the complexity of the library? That also, I think, is a function of the actual input material sample. Since right now we are trying to reduce the input, sample with an improved GLORY condition, so we actually haven't, come up, let's say, a recommended sequencing depth for a given amount of the input sample. We don't have that curve at this moment. I think the current priority for us is to further improve the condition so that it gets milder, it gets, preserve the longer fragments of RNA so we can do more possibilities. So we're sort of focusing on that.
I might say that there might be different buckets of questions. So one question could be, where are all the high stoichiometry M6A sites? And by high stoichiometry, let's say above 70% methylation. And there you might not need, more than 40 or 50 reads. But then another question is, which sites are changing in their methylation? And if your site is changing from 40% to 60% methylated, that you may need enough sequencing depth to be confident that that is not sampling noise. And so there you might need to be deeper. And then the third bucket is assigning an M6A load per transcript. If in a lot of cases, when you use these requirements of 50 reads per site, you're only going to be able to assign a load to maybe the top 30 or 40% of abundant genes. And so, and assigning that load is important for understanding the degree to which that transcript might be affected by methylation. So I think there are some cases where you do want high enough read to answer some of the latter two questions. But if you're just saying, is it methylated? I think we could have lower numbers of reads. I don't know what people think.
Well, that's where I think, ETAM, Weixin was talking about ETAM Sanger can be really advantageous too, because you could use GLORY or ETAM to get the transcriptome-wide information. But then, like you said, Samie, if you have transcripts of interest that you really care about or a certain molecular pathway, why spend the money on a transcriptome-wide experiment when you can do it in just a targeted approach? So that's where I think, coupling ETAM with just the target specific Sanger sequencing can be really powerful, because we do a lot of that in our lab too with DartSeq, and we're starting to try and use ETAM for that purpose as well.
Well, I definitely agree that it's important to validate any predicted stoichiometry change using maybe the ETAM Sanger. We actually do GLORY Sanger, Chengqi. You said you can't do it. We do it. We actually can sequence and quantify the number of reads.
I think the successful rate is not as high as ETAM-seq in our hands. That's why I'm not, promoting it like 100%. But yes, certainly we can, get the Sanger data from GLORY as well.
It doesn't work every time, but it works sometimes. But I think, I thought your answer to Schragi's question was going to be this, which is my view of how to get greater depth is not to sequence in greater depth, but to have more independent biological replicates and then sum up from the individual biological replicates to make your measurement of the stoichiometry. I don't know, Schragi's shaking his head, and I definitely want Weixin to jump in on Schragi's question. But Schragi, don't you think that's the answer? Just more replicates, even if don't sequence deeper, you're just going to get duplicates. Do different biological replicates.
I wasn't shaking my head. No, I was thinking about it. I think to some extent it can work. We've tried it now a couple of times. There's also batch effects you need to start to worry about. We've certainly seen these when you when you treat things side by side, you start seeing these differences. And there's also probably ways to overcome these as well. It might be part of the solution. I think in general, if it's always better to have more replicates and to have more depth, I'd say, in any context, you can always pull replicates together, right? But you can't do the same for depth. So I'm always for that kind of solution. But the question is, will that solve all your problems? Perhaps it might help you overcome some of them. I'm not sure this is going to help you solve everything you need here. And it certainly increases the workflow, right? I mean, instead of having to do it for 2 samples, you now suddenly have to do it for 50 samples, right? That certainly changes things.
Yeah, I'm really afraid that a lot of people are going to use ETAM-seq and GLORY and then look at big differences in M6A, but it's only from 20 reads or a small number of reads. They're not going to use the right filters to really make conclusive validations. I mean, I think Chengqi brought up a 50 nucleotide read threshold. That's great, but I hope everybody, shows that their dynamics come from lots of read depth and occurs across samples and hopefully can be reproduced by ETAM Sanger or by GLORY Sanger.
Right. So we do have a recommended coverage, I think, in the paper. So that's one thing. And also, I want to add one additional piece of thought. I like the idea Schraga and Samie mentioned about it. It's not perhaps the overall methylation of let's say one mRNA or one gene that actually really matters. That it is perhaps more important than individual MC sites. Yes, I agree with that. But I would also like to think that in particular cases, let's say different modification sites on the gene depends on the location. Let's say 5' UTR, CDS, or 3' UTR. Whether or not different MC sites could have different functions, I think that's also one new hypothesis. If we just average everything up, right, then we probably don't see a global methylation change, but that doesn't necessarily correlate with function. So I would still, I agree with you that certainly the global methylation level is important, but I would still want to argue that these, ability to look at single base M6A level could, let's say, prove or disprove one hypothesis, functional hypothesis.
Yeah, I agree. And I mean, that's been shown, right? That that where an M6A site is can influence the function of the site. So I completely agree with your point, Chengqi. I think, maybe not just a whole RNA level methylation, but region specific methylation. I mean, of course, then you get into what's the size of the region then, and what if it's in the 3' UTR but overlaps the coding sequence? Does that matter? Do we use those boundaries? Do we use strict, exon boundaries, things like that? But I think, our thought certainly has shifted from the importance of individual sites to the importance of regions of methylation within a transcript. And I think we've learned this morning from everyone's talks that that's kind of becoming more what we're realizing might be important, right?
Well, I think these new methods can really help us address, this idea of whether, let's say, 5 clustered M6A sites are different from 5 widely spaced M6A sites, and they have different biologies and things like this. In the last few minutes, I want to switch to Schraga, and, your presentation really highlighted something that, I've been a fan of, and people may have read our review in Molecular Cell, where we argued that there's probably not so much dynamics of M6A because, what we had argued and you guys have proven is that genomic architecture can determine, has a big role in determining where methylation can occur and where it doesn't occur. But in light of that, in normal scenarios, whether it's differentiation, and I know you studied this in your Master Seek paper, but differentiation, neurodegeneration, what's the prospect for site-specific dynamics of methylation? When would it happen, or does it not happen? Because if one way to read your paper is the M6A profile is fixed, and just use my program M6A pred 2, and you'll know where the methylation is. What's the role, from your perspective, of dynamics? And should we even be studying dynamics anymore?
No, we should definitely be studying dynamics, and we're studying them ourselves. And you should not believe M6A pred 2 for this. For this, you should use ETAM-seq and Glory and figure or DART-seq, right, and actually measure to what extent things are dynamic. But yes, I think with M6A pred 2, the way I think about it is one should think about it as a first approximation. So if one wants kind of a first approximation, a baseline of what shapes M6A maps, I think to a large extent, kind of the simple rules that we define, regardless of the mechanism, but it just they're the ones shaping the overall topologies. And I think there's a lot of work, including some of also Chengqi's earlier work in M6A, where we looked at it in about a bunch of mouse tissues, our earlier work where we looked at it in different tissues. It's all kind of converging that to a large extent, the landscape is fixed, right? To a large extent, but then there's always the outliers, as also Kate was talking about this morning. In terms of, why and how M6A would be dynamic, first, what we describe is mainly a model of what we think impacts M6A biogenesis, right? But M6A steady state levels are more than just the onset of M6A. They're also about M6A being recognized and read and degraded and so forth. So any source of biological variability that could give rise, for example, to differences in readers, leading to differences in their ability to recognize and perhaps degrade M6A containing RNAs, right? That would be a source of differences in M6A profiles across tissues, right? So even assuming that everything that has to do with the position of M6A is entirely and uniformly fixed across cells, I could still imagine scenarios whereby the actual steady state M6A levels might change as a consequence of things that happen down the road.
But Schraga, in that statement, I think you would argue that there would be global up and global down, not site-specific up and down. You know, is that right? Is that what you're getting at? Because I that's what I would tend to believe, you know, and I think ETAM-seq and Glory can really tell if you compare two tissues, if globally there's a, you know, increase by 5% at all the sites, you know, on average. But are you arguing for site-specific? Because that's where we see so many papers that say, oh, this site in this transcript went up dramatically. They didn't use ETAM-seq or Glory, so they don't report that it went from 20% to 80%, which now the methods are available. And I hope in the future, every paper gives us the exact percentage before their stimulus and exact percentage after, because we have the methods to do that now. But what you're saying speaks to global trends, not site-specific, right?
More precise. I can think of three different resolutions at which I can think of dynamics. One is exactly this, site-specific levels. And I must say, you know, there's always the question of demethylases and how relevant they are, but Chengqi now showed, you know, with FTO, it's for me the most compelling set of plots I've seen to date about actual global sites being, you know, identified, you know, that are being induced upon FTO knockout, right? So I think, you know, who knows? I don't know what the code is there and to what extent these might be specific and what determines the specificity. But, you know, assuming these things happen and demethylation could potentially be happening in a site-specific way, then that's exciting, right? And that could be one way and this could be achieved. I think another level at which it could be achieved would be global. As you said, you know, it's basically you have more of this, you have more of a reader, you have more of a writer, all of the M6As get impacted in a similar way. What's most interesting to us, and it's something we've been working on, is could this be happening at the gene-specific level? So let's say something happens to the metabolism of a gene, right? Let's say it's suddenly becoming more nuclear, it's becoming more cytoplasmic. Let's say the gene itself is susceptible to regulation. How would that impact M6A levels along that gene, right? And I think in that case, you might not expect it to impact an individual site here and there in the gene, but you also wouldn't expect it to be global, right? You would just expect it to impact the different sites that happen along the gene.
That's interesting.
So that's something we're thinking about a lot. We have some things that are in support of this. We have others that contradict it. So it's work in progress at the moment.
All right. We should really wrap up. I apologize. So we do have a lunch break, and you know, maybe some of these topics may come up with different speakers in the second panel. You know, a lot of you have asked questions, and the speakers will try to address them in the question and answer box. I want to remind people it's 1 a.m. in China for Chengqi, so he may not be able to do it, but we'll see. But thank you, everybody, and we're going to reconvene in 25 minutes after the lunch break. And so that's 1:30 p.m. Eastern time, so 25 minutes, and we'll see you then. And Kate will take over for the nanopore and the new nanopore technologies for M6A. We'll do nanopore for other modifications on Tuesday. All right. Thank you, everybody. We'll see you soon.